Hi everyone, thanks for coming to my talk. My name's Jim Barnett. I'm a postdoc at McMaster University where I work on animal coloration. Today I'm going to be talking about some of my recent work into how human disturbance may affect the visual ecology of poison frogs. So animals use colour for a huge variety of different functions, and I just want to illustrate that with some of the different species of amphibian that we've been working on in our lab recently. So we have some species which are using camouflage, with cryptic patterns that blend into their surroundings, and others which are using these bright, conspicuous patterns either to warn predators away or communicate with conspecifics. In both cases, if we really want to understand how this colour works, we need to know both about the colours themselves, but also about the context within which that signal has evolved and where it is viewed by observers today. Now, that natural context within which animal coloration has evolved can now be under threat as human activity changes the characteristics of the visual environment. We were interested in seeing how such changes may affect a variety of different colour patterns found within the same environment and being affected by a single change caused by human activity. To do this, we looked at three very different frog colour patterns found in the Bocas del Toro archipelago in Panama. We looked at the brown and beige stripes of the non-toxic Allobates talamancae, which looks like quite a camouflage pattern. And then we looked at two different colour morphs of the poisonous frog, Oophaga pumilio. We looked at a green and black spotted morph, which is generally considered to be cryptic, and then also at a red and black colour morph, which is considered to be conspicuous and aposematic. Now, even though these three frogs look quite different, they do share a lot of similarities in their behaviour, their morphology, and their ecology. They're all about the same size, they all have similar lifestyle, being diurnal and eating a diet of small invertebrates, and they live amongst the leaf litter of these tropical rainforests. So under natural conditions, these three frogs will look something like this against a natural substrate of dark brown leaf litter. Human activity, when it comes into a tropical forest like this, quite often removes some of those big trees which are casting a shadow over the forest floor. Without those big trees and the canopy cover above, we get more light onto that forest floor, and that encourages the growth of small plants within that leaf litter, leading to a general greening of that forest substrate. So in areas of increased human disturbance, we may find the frogs are more likely to be viewed against a background like this. So we were interested in investigating how those changes to the visual environment may affect the detectability of these different frog phenotypes. To do that, we took calibrated photographs of these frogs and the backgrounds in the natural and disturbed habitat, and then we used computational visual modelling to assess how closely the colours of the frog match the colours of the background. Once we're able to quantify the amount of distinction between the colours of the frogs and the backgrounds, we wanted to know about how that would affect detectability. To do that, we used human participants as surrogate predators and presented these crops of the dorsal pattern of the frog against both the natural and disturbed habitats. We then recorded how quickly our human subjects were able to find these frogs as our metric of detectability. So what did we find? Let's have a look at the visual modelling data first. All of these data I'm going to show right now come from a model of a predatory bird visual system, but the general trends are also found in a snake visual system, in a poison frog visual system, and in a human visual model. So on this plot here, we're comparing the main colours of these three frogs, as the dark brown, the bright green, and the bright red, against the colour of that natural dark brown leaf litter. On the y-axis, we have a measure of chromatic contrast, which is equivalent to JNDs. If you're not familiar with those, all you really need to know is that the higher a point is up on the y-axis, the greater the distinction between the colour of the frog and the colour of the background. So under natural conditions, we can see that the red frog is much more distinct from the background colours than either the brown frog or the green frog, which are both quite close matches to that natural background. In the disturbed site, we see that things change a bit. The red frog is still highly contrasting against that background. But the really interesting change that we see comes from looking at the two cryptic patterns. In this disturbed site, compared to the natural site, the green frog is now a closer match to that substrate, whereas the brown frog is now more distinct from the disturbed background than it was from that natural background. So we may expect that this would affect the efficacy of any camouflage that these frogs are relying on. So from the visual modelling, we can say that there are changes in chromatic contrast between the two different backgrounds for each of our three different frogs. We now wanted to see whether those changes actually affected the detectability of these different phenotypes. This is where our human detection experiment comes in. And now again, we're comparing those three dorsal colours against that natural dark leaf litter substrate. Our y-axis here is now reaction time. So unlike in the last one, now if you go further up on the y-axis, you're getting towards better camouflage. It's taking longer for our participants to find the frogs. And in this natural environment, we find that the brown frog is indeed more cryptic than the red frog, but also the green frog, which are much more detectable. Now you may spot the discrepancy here between what we predicted from our visual modelling data, where our green and brown frogs were both a close match to the background. Now we have some data which is presented in the paper, but I don't have time to go in here. But really this distinction comes down from the fact that these black spots are actually quite distinct from the background patterning, and so it makes the frog detectable even in the absence of colour. But the important thing for our question here is the comparison to the disturbed habitat, and there we do find the trend that we would expect from our visual modelling. In the disturbed site, the red frog is still found very quickly, but the green frog is now found more slowly than it was in the natural site, and the brown frog is found more quickly. So in terms of camouflage, the brown frog has become easier to find, and the green frog has become harder to find. So to summarise, we found that human activity is changing the characteristics of the visual environment, and that change is having an effect on different phenotypes, but those effects can go in different directions. So we think these community-wide changes may have knock-on effects to predator behaviour, as different prey types may change in their apparent availability as their relative detectability changes. So I'd like to say thank you very much. If you have any questions, you can reach out to me via email or Twitter. Thank you very much.